Shall I start?
Yes, madam, may start. It's time now already.
Very good afternoon, all dignitaries. Myself, Professor Dr. Subhasri Sahu, as Organizing Secretary of International Conference on New Horizons in Pharmaceutical Research 2021, ICNSPR 2021, organized by KANAK MANJARI Institute of Pharmaceutical Sciences, Chain, Rourkela, India, in association with Indian Society of Technical Education, ISTE, New Delhi, India. I must introduce the session to before we introduce the first speaker. Let the audience know about the session is about how it relates to or differ from other sessions at the conference and how it is going to be take place. By introducing the session, we are providing the audience with a global view on pharmaceutical research that will help us assimilate the details. By making the participants feel welcome, we also incline them favorably towards the speakers. Our first keynote speaker is going to discuss about lipid-based drug delivery system. The principal objective of formulation of lipid-based drugs is to enhance their bioavailability. The use of lipids in drug delivery is no more a new trend now, but is still the promising concept. Lipid-based drug delivery systems are one of the emerging technologies designed to address challenges like the solubility and bioavailability of poor soluble drugs. Lipid-based formulations can be tailored to meet a wide range of products requirements detected by disease indications, route of administration, cost consideration, product stability, toxicity, and efficacy. These formulations are also a commercially viable strategy to formulate pharmaceuticals for topical, oral, pulmonary, or parenteral delivery. In addition, lipid-based formulations have been shown to reduce the toxicity of various drugs by changing the biodistribution of drugs away from sensitive organs. However, the number of applications of lipid-based formulations has expanded as the nature and type of active drugs under investigation have become more varied. This theme mainly focuses on novel lipid-based formulations and their prominent applications in pharmaceutical drug delivery. Our second keynote speaker will discuss about new methods of identifying counterfeit drugs. Owing to the numerous consequences of fake drug products, various methods have been devised for testing counterfeit drugs. The principal requirements for these methods is to identify the active pharmaceutical ingredients, API, and substances other than API, and excipients, additives contained in the dosage form. We can get a clear picture after second speaker's session about the counterfeit drugs, identification of counterfeit drugs. After that, our participants all over the globe are going to share their novel ideas regarding pharmaceutical research. So now I request Professor Dr. Manoj Kumar Dalai sir, Organizing Director of this International Conference ICNSPR 2021, to welcome our first keynote speaker. Over to Professor Dr. Manoj Kumar Dalai sir. Sir, please.
Thank you, Subhasri madam. Namaskar. Good afternoon, everybody. Today, we have the invited speaker, Professor Dr. Kunal Pal, who will going to deliver the topic liquid-based drug delivery system. Madam already describe the details of this area. So I think most of the pharmacy students are going through this topic. So before starting of this session, I like to introduce Dr. Kunal Pal. Dr. Kunal Pal, Associate Professor of Biotechnology and Medical Engineering Department, NIT Rourkela. He did his Master in Engineering from Jadavpur University and PhD from IIT Kharagpur. He has been more than 11 years of working experience in the field of biomaterial therapeutic delivery system and tissue engineering. Also, he has vast experience in the field of medical electronics and instrumentation. He had undergone a project reviewer for the Israeli Ministry of Health, particular the topic food and nutrition implication on human health program. That program was carried out on the year 2016. He has already guided a 12 number of PhD students and also number of PG students. In his credit, 200 research articles and many book chapters already published. He is well cited researcher having more than 2000 citations. So with this introduction of Dr. Kunal Pal, now I am requesting sir to say a few words and enlighten this particular topic to our participant. So please, Dr. Kunal Pal sir.
Yeah, thank you, Professor Dalai, for the nice introduction. So without wasting time, I would start my presentation. Professor Subhashri, can you see my screen?
Yes, sir. Yes, sir.
Yeah. Okay. Thank you, Professor Vera. Yeah. Good afternoon, everyone. So I am Dr. Kunal Pal, as introduced by Professor Dalai, and he has given a nice introduction. So I need not to speak much about myself. So today I'll be talking about our recently published paper, effect of carboxylated carbon nanotubes on physicochemical and drug release properties of oleogels. Okay. Now, many of you have heard about this nanotubes, carbon nanotubes. So in short, they are regarded as CNTs also, right? So many researchers, what they are doing, they are functionalizing these CNTs to get different functional groups over the CNTs. So one such group is carboxylation. So if we functionalize the CNTs with COOH group, that is the carboxylic groups, then we get the carboxylated CNTs. Now there is another new term most of you would find over here, and that term is oleogels. Okay. Now this oleogel, it is being derived from two words. The first one is oleo and the second one is gels, right? Now oleo, it means oil, and gels, it means any three-dimensional structure which can entrap any liquid solvents. Okay. So its counterpart, one of the counterpart is hydrogels. Okay. And many of us are quite conversant with the hydrogels. What are the hydrogels? The hydrogels are basically the 3D structures which can hold water within its structure. Okay. So oleogels, they are a counterpart of the hydrogels. What is the basic difference? The basic difference in oleogel, we have oil In hydrogels, we have water molecules as the liquid phase. Okay. Till date, you will find a lot of research has been carried out on the hydrogels. Why? Because if you look into our human body, what happens over there is that in our human body, we have 70 to 80% of water, and all the matrices which are present within our body, say the extracellular matrices or the organs, if we consider them as the 3D structure, they have a large amount of water. So till recent past, all the researchers, many of the researchers, they have been concentrating on the hydrogels. Okay. But there is another aspect within the tissues or the cells. What are those? Those are, when we consider about a cell, okay, cell, it has got a large fraction of water, no doubt, true. But the same cell, it would have some phospholipid bilayers. Okay, you will find this kind of bilayers, and the internal structure of this bilayer is lipid based systems. Okay, so if you see our human body, it nicely houses both hydrophilic and hydrophobic components within our body. Okay, and this is an important thing. You will find in many cases when there is a low bioavailability of the drugs, we talk about improving the permeation of the drug through the topical route into the human body by using some lipid based or oil based systems. It may be liposomes. Okay, it may be emulsions. It may be nano emulsions. Okay, and there are many. So there are numerous products, so I cannot talk about them. Another important product is solid lipid nanoparticles. So if you see, since last 20 to 30 years, people has been working towards the lipid based system to improve the bioavailability of the drugs within the human body. That's good. But there is one problem associated with these all types of formulations. How do you prepare these formulations? The preparation of these formulations is quite hectic. Okay. Apart from that, there are stability issues. Okay. So you will be finding that, yeah, say the liposomes, you will be finding it very. If you want to make the liposomes, you have to use a very complex systems. If you talk about the emulsions, they are inherently what you call unstable in nature, right? When you talk about nano emulsions, they are inherently stable, but they would require a high amount of surfactants within it to prepare the. Okay, solid lipid nanoparticles. You need a high and a number of high-ended equipments to synthesize those SLNs, okay, solid lipid nanoparticles. So if you see, most of these lipid-based drug delivery systems, they have their own disadvantages. So in this regard, in the last decade, I mean the last 10 years, you will find that researchers have been looking towards this oleogel-based system. Now, interestingly, when we talk about this oleogel-based system, till recent past, since our group, they started working on oleogels. Before that, these oleogels used in foods. For food application, people have been exploring. Our group, since the 5 to 6 years time, we have started working on the oleogels, and now you will find many other researchers across the globe, they are working towards the development of the oleogels for drug delivery and other biomedical applications also. Okay. So I am happy to tell you that our group, it is the fourth largest group across the world who are working in the field of oleogels, and the leader in the field of oleogel is Professor A.G. Marangoni. Okay, and he is from University of Guelph, Canada. So he is a very good researcher with and he is a prolific researcher, and if you want to learn more about oleogels, I would suggest you please go through the papers of Professor A.G. Marangoni. Okay, he mainly works in the field of food science. Okay, now what is the advantage of these oleogels? The first thing is, as I told you, it is a lipid based system. Okay, what is the second thing? As compared to the other lipid based systems over here, liposomes, emulsions, nanoemulsions, SLNs, you can prepare the oleogens in a relative simple manner. Okay, now what happens? Due to this reason, since we can synthesize oleogels in a simplistic manner, we can upscale the production of these systems in a better way. Okay, in our recent reports, I have not included on the other ones, where we have found that these oleogel based systems, they can improve corneal permeation. Since many of you are from the pharmaceutical background, you know that delivering drugs within the eye, it is a challenging perspective, right? So oleogel can provide a tool to deliver drugs or to improve the bioavailability of the drugs within the internal structures of the eye. How it will do? It can improve the corneal permeation. So if you have an improved corneal permeation, you will have an improved drug bioavailability at the site of action. Okay. So with this small introduction about what you can expect in this lecture, I would proceed further with the actual topic. And as I said that we have published this paper, and this paper has been published in Colloids and Surfaces A, Physicochemical and Engineering Aspects. It has been published recently. Okay. Over here we had Professor M. Serecuia. He is from Portugal. I thank him to be a collaborator for this project, and he has helped us in many ways. So coming to the background, so oleogels, they are defined as edible oils that are immobilized within thermoreversible and three-dimensional networks of oleogelators. Okay, I said, if you remember over here, these gel-based systems, what are these gels? Now prepared of, these gels are prepared of gelators. Okay, now the 3D matrix of the any gel, which is used to synthesize these gel-based systems, they are regarded as the gelators, and they are the solid phase. Okay, and within this network of this solid phase, you will be having the solvent phase, which I am drawing as the red dots. Okay, so when we talk about oleogels, we need these gelators, but since these gelators, they are hydrophobic in nature, they are regarded as organogelators. Okay, and if you are using oils, in this case, since we are using oleogels, so in that case, the organogels are regarded as oleogelators. Okay. Now, quite often you will find that organogelators and oleogelators, they are interchangeably used. Oleogelators are mainly used basically if you're using any type of oil, okay, if you're immobilizing any type of oils, then it is called oleogelator. And organogelator, it is a vague term. It can be used to immobilize oils. It may be used to immobilize the organic solvents, right? So this is a more generalized term. This organogelator, it is a more generalized term. Okay. So as I said, over the recent years, there has been a continuous interest of the researchers for developing controlled drug delivery systems using these oleogel based systems. Another important advantage of this system, what I forgot to mention in my introduction, is that since you are using with oil, these oleogels, they are resistant to microbial contamination. Because if you see, if you have a bacteria and if you make a hydrogel based products, the hydrogel based products, it will have an aqueous environment. If you use polysaccharides, then the bacteria and the other microbes, say the fungi, they can use those polysaccharides or the proteins, and they can use it for their growth, and and they will start growing onto the formulation. So that is one of the major problems with the hydrogel based systems. Whereas in oleogels, you will find many of the microorganisms, they will not grow. Okay. And due to this reason, these oleogels, they are resistant to the microbial contamination. Another important thing is there. These oleogels, they have not only been used to deliver lipophilic bioactive agents, which includes curcumin and beta carotene, but also it has been used this hydrophobic for delivering the hydrophobic drugs and also the hydrophilic drugs. Okay, so you can see it can when I say this lipophilic bioactive agents, this curcumin and beta carotene, they are basically the food grade agents. Okay, and when we talk about drugs are some molecules which have synthetic molecules which have some activity against and they help us in in some pathological conditions. Okay, whereas the bioactive agents, they are some of the they are the chemicals which are generally available in the nature, and they can improve the biological activity within the human body. So there is a subtle difference between the bioactive agents and the drugs. Okay. Unfortunately, at present, the research on the use of vegetable fats as the structuring agent of the oleogels is still in infancy. When I talk about vegetable fats, most of you who are from the pharmaceutical background must have heard about cocoa butter. Okay, but it is being used in various formulations like SLNs. It has been used in suppositories, right, and so on. So there is a problem with this cocoa butter. The cocoa butter is quite expensive in nature, and this limits its use in low cost formulation. Okay. However, apart from this cocoa butter, there are not much vegetable fats which are used for making the formulations related to the oleogels. In this regard, we have a lot of fats which can be obtained from various agricultural wastes. Say for example, when we eat mango, we have these mango seeds, and if you break open the outer shell of the mango seeds, you will get the embryo, and from this embryo you can extract fat, and that fat is regarded as mango butter. Okay, our group is also involved in utilizing this mango butter, because this mango butter, it is being extracted from the seeds, which is a waste. So you will find the price of the mango butter is quite cheaper as compared to the cocoa butters. Okay. Like this mango butter, there is another type of fat which is regarded as the shea butter. Okay. And this shea butter, it is obtained from the seeds of the West African shea tree. Okay. Its scientific name, it has been given over here, Vitellaria paradoxa. Okay. Now, say this mango butter or even the shea butter, people means the researchers, they are trying to explore it as an alternative to cocoa butter. Okay. In other words, we can say this mango butters and shea butter, they are cocoa butter alternative, okay, or cocoa butter equivalent, and hence it can be used as cocoa butter equivalent, cocoa butter alternative agent. Okay. So why this is so? This is because both mango butter, shea butter, and there are certain other fats like lychee fat is also there. These fats, they have similar physicochemical properties which is similar to that of cocoa butter. Okay. And due to this reason, these naturally derived fats from the vegetable sources, they are gaining importance in the food and the pharmaceutical industries. Okay. Now, why these fats, they have similar type of properties? Because the fat content over there, I'm sorry, because these fats, they have similar fat content as that of the cocoa butter. Okay. Now, in shea butter, which we will be using over here as the main vegetable fat, over here we have a major component which is regarded as the 2-oleol distearin. Okay. So it is an derivative of stearic acid. Okay. And as I said that many fats, they have been used to develop various fat-based delivery systems. It is true with the shea butter also. The shea butter, in combination with other vegetable oils and surfactants, they have been explored to synthesize emulsions, lipospheres, microparticles, and solid lipid nanoparticles. Okay. Till date, we did not find any literature that has been used to develop shea butter to produce the oleogels. So that gave us an enthusiasm to try and use the shea butter to produce oleogels. Now, there are several edible oils in the market which has been used not only for the food purpose applications but also for the pharmaceutical applications. Those includes palm oil, olive oil, flaxseed oil, safflower oil, and so on. Okay. And these oils, they have been used for the preparation of the oleogels. Okay. Among the above mentioned oleogels, this wheat germ oil, it is becoming vividly popular in developing countries because it is having very good health benefits. Apart from that, it is safe for consumption and it is having nutritive properties. And if you see, wheat, it is very commonly grown in India. Okay. And it is it can be extracted from the wheat germs at a very lower price. Okay. And as I said, see, this wheat germ, it is a byproduct of the wheat milling industry. So what you are doing, you are using the waste and valorizing it to make a value added product. Okay. In short, it is called VAP, value added product. Okay. Recently, some research groups, they have used this Vidgem oil for preparing controlled drug delivery systems like emulsions, Pickering emulsions, and nano emulsions. Okay. Further, when you alter the composition of the shea butter or the fats and the oils, then we can alter the crystallization properties. Apart from that, we can also alter the crystallization property of the fats by adding some this crystal modifying parameter substances, so, say for example fats, emulsifiers, free fatty acids, and waxes. Okay. In this study, in our case, we have explored the use of CCNTs, carboxylated CNTs, as a crystallization modifier of this of the shea butter, right? And if you see CNTs in its pristine form, it has been explored as the crystallization modifier for 3 different types of triglycerols. Okay, so we have taken the inspiration from here, and we thought, okay, if we use these carboxylated CNTs, can we alter the crystallization properties of the shea butter so that the release properties can be governed? Okay. Now, the CNTs have been investigated as drug delivery carrier owing to its unique features like high aspect ratio, large and functionalizable surface areas. Okay. Further, in comparison to the pristine CNTs, the carboxylated CNTs, they possess very low toxicity. So, taking consideration from this discussion, we have used shea butter and we have used Vidgem oil to develop oleogels. And then within this oleogels, we have added CCNTs in different proportions so that we can alter the properties of the oleogels by altering the crystallization properties of the shea butter. Okay. So we will see in the next slides how the CNTs, they can manipulate the crystal structure of the shea butter within the oleogels, and how we can have uh, different types of oleogels where we are able to control the release of the drugs from these oleogel based systems. Okay. So when we started the experiment, we first found out the critical gelation concentration. I hope everyone knows what is CGC, critical gelation concentration. So it is the lowest concentration of the gelator molecules to induce gelation of a solvent. In our case, what is the gelator? The gelator is the shea butter. And what is the solvent? The solvent is the Vidgem oil. Okay. So we found out the CGC, that means the minimum amount of shea butter required to induce gelation of the Vidgem oil. Please be reminded, for specific fat oil combination, this CGC would differ. Okay. So this constitutes that the CGC of shea butter to induce gelation of WGO was 60%. Now, in the next formulations, what we have done is that we have kept this composition constant.
Sir, you are muted. Kunal sir.
Oops.
Yes.
I don't know how it got muted. Sorry. Yeah. So when we talk about this, what I was telling that when we prepared these drug-loaded systems, in the drug-loaded system, we have added 50 mg of ciprofloxacin hydrochloride within the system. Okay, and the codes for these formulations are this one, SW1D. Okay, so anywhere you will find that at the last letter is D, that means it is the ciprofloxacin loaded what you call oleogel based systems. Okay, so these are the pictographs of the oleogels. So if you see, the first one is the SW1, so it did not have any oleogel and any CNTs, so it appears as bright yellow in nature. When we added small amount of carboxylated CNT over here, so you see the brightness of the yellow, it has reduced and it is appearing in somewhat dark. In SW3, we had a greenish tinge. Okay, whereas in SW4 and SW5, you will find that the oleogels are completely black. Okay, now these pictographs are for the inverted from the inverted tube test method for the formulation, which allows us to understand whether a gel formulation has been formed or not. So when we have inverted the tube, if you see, all these formulations, they are not flowing under the gravitational force. That means all of them, they are maintaining their structural integrity. Okay, since they are maintaining the structural integrity, that means they are relatively stable gels and they may be analyzed for further studies. Okay, now if you see the this pristine oleogel, you will find that there are certain globular structures which are present all throughout the matrix, and these globular structures, they are joining with each other and they are forming a network-like structure. And when they are forming this network-like structure, within these spaces, if you see, there are some bright spaces over here. These bright spaces are for the oil. So in this case, WGO. Now, when we have incorporated CNTs within this system, if you see, there has been the agglomeration of this system, but still they are forming a network-like structure, and you will find some oils are there. And if you see, there are some black dots throughout this microscopic structures, and these black dots, they are basically the agglomerated CNTs. Now you would say, sir, then you might not have prepared the samples in a proper manner, right? But the thing is that we have to consider that CNTs, since it is carboxylated form of the CNT, it is hydrophilic in nature. Okay. During the preparation, we have taken all the precautions, we have done the homogenization, and still we have found that these black dots, they are remaining. So why these are remaining? If you see that if you take an oil and if you put a drop of water into that, it will form a agglomerated system, okay, of the water droplet within the oil system. The similar thing has happened over here when we have incorporated these hydrophilic carboxylated CCNTs within an hydrophobic phase. These hydrophilic systems, they have tried to form integrated structures, or they have tried to form these agglomerated structures, and they are present all throughout the matrices in an even way. Okay. Now, when we increase the concentration of CCNTs in SW3, so if you see over here, agglomerated structures of CNTs, they are there. And apart from that, if you look into this fat architecture, there has been a complete aggregation of this this thing. That means when we are incorporating CCNTs within this fat system, it is inducing a change in the crystallization process of the within the WGO system, and it is totally altering the properties. When we went for this SW4, see, further there are some aggregations which are there, and if you see the agglomerated CCNTs, they are also present. Finally, if SW5, so what happens over here? If you see these oleogels, these oils and these fats, they are present. If you see, both of them, they are present. You are having almost 50-50 darker phase and 50-50 lighter phase. Okay. So that means these systems, they are forming bicontinuous system. Bicontinuous means both the fat phase and the oil phase, they are present as the continuum phase. Okay. In the other cases, in SW1 to 4, you will find that the network structure is being formed by the fat molecules, and the WGO phase, it is present within this fat structure. Okay. However, in SW5, there is a complete change in the architecture, microarchitecture of the samples. So you can predict from here that from SW1 to SW4, you can see some pattern in the characterization properties of the systems, but in SW5, the property will be very different. Okay. So we have got an idea from here. So then we went for the further evaluation processes. When we look into the FTIS spectroscopy, this is the FTIS spectroscope of the shea butter. This one is for the this Wilczynski oil. Okay. Now, if you see over here in shea butter, there is a peak at around 3400, 30 centimeter inverse of wave number. So this peak is basically due to the hydrogen bonding. Okay. In oil, since it does not form a solid structure, this over here we are not able to see the peaks. Okay. If you see, when we are having lowest amount of when we are having this oleogel, if you see, we are having a very strong intermolecular hydrogen bonding, even higher than that of the normal or the pristine shea butter. Now, as soon as we started adding the CCNTs, if you see, this peak is diminishing. Okay. That means with the increase in the CCNTs, the inter and the intramolecular hydrogen bonding within the oleogel formulations, they are decreasing. Okay. I'm not going into the other peaks because all the other peaks, they are common. If you find all these peaks, they are common in each of the fat-based systems, because they are hydrocarbons in nature, and all of them, they will show the similar pattern. Okay. So the main change was in this area. Now next, and if you look into the XRD analysis, we got a very sharp peak at around say 24 degree to theta. Okay, and this is the most sharpest peak, and then we had the other peak. So this one was for the SW1, and as we started increasing the CNT concentration, CCNT concentration, you can see there is a variation in the peak height of all of this. Okay, now if you see the intensity profiles for each of these case, we have kept the same scale so that you can analyze the changes in the peak height in a proper manner. Apart from this strongest peak, there were certain other peaks which were also there in all of the formulations. So these peaks were there, but there were subtle changes in the peak positions, which we'll be discussing when we'll be discussing the results through the in the tabular format. Okay. Now, after getting these peaks, what we did is that if you see, there are a certain number of peaks which are available within the systems, but they are overriding over each other, right? To separate or to segregate the peaks, we need to do deconvolution, and then we did the deconvolution, and these are all the deconvoluted peaks, and the deconvolution can be performed using the origin software. It can be done using the origin software. Okay. Now, once you get these individual peaks, see, once you get these individual peaks, we went for the determination of the various properties like the d-spacing, crystallite size, lattice strain, dislocation density. So d-spacing, what is d-spacing? It is the interplanar spacing between the crystals. Okay. I hope everyone knows the basics of the XRD. So if not, please let me know. I can discuss if anyone does not know. So I can discuss in brief. Okay. So over here, if you look over this d-spacing, it was 4.582. Then here in SW2, it was 4.572. Then 4.514, 4.571, 4.598. So if you see, there was not a significant change. There was no significant change. Okay. However, if you look into the crystallite size, which is not only dependent on the peak position, d-spacing, it is basically dependent on the peak position only. Okay. Whereas the crystallite size, it is dependent on both peak position and the FWHM. Okay. So when we calculated this, if you see the crystallite size of SW1 was 11.756. When we incorporated CCNTs in the lowest concentration, it was 17.903. That means the crystallite domains has increased. That means the crystallite size within the fat, solid fat, it has increased. However, in SW3, there was a reduction. Interestingly, in the case of SW4, it achieved a critical level and there was a significant increase in the crystallite size. Again, in SW5, since there was a significant decrease, in the crystallite size, and this was quite expected because if you look into the microscopic picture, I have already told you that we can expect a sudden decrease in the architectural or sudden change in the properties of the SW5 system. Okay, so that's what we are seeing over here. If you see, there was a drastic decrease in this in the property of this one. Okay, now talking about the lattice strain, if you see the lattice strain of SW1 was 0.027. It was nearly the same in over here. In SW3, the lattice strain was quite high, and this can be reason to this lower crystallite size. If there is an increased lattice strain, then the crystals will not be able to grow to larger crystals. If you see in the SW4, the lattice strain, if you see over here, what is happening? It is the lowest. It is the lowest among all these formulation. So it was bound to have a larger crystallite size. Are you getting my point? Then again in this SW5, there was a quite high lattice strain, and it was quite expected, because in bicontinuous system you do not know what is the continuum phase, whether it is the gelator phase which is the continuum phase, or is it the oil phase which is the continuum phase, and there will be some problems over here there. Okay. So this dislocation density, it is related to the lattice strain. So I will not discuss this one over here since we are running out of time. Then we went for crystallization studies. So we studied how the sample's temperature, it changed with respect to time, and this experiment was done at 25 degrees centigrade. Now if we see, there was an decrease in the temperature profile, but suddenly at around 13 degrees centigrade there was an exothermic reaction, and again there was an decrease. And if you see, it is not just an one-off phenomena. This phenomena was present in all the other CCNT containing systems. Okay, there was certain change in the peak positions which we will discuss in the next slide. So why this is happening? Because we are not giving external thermal energy, because we are doing the experiment in at an isothermal condition. So what is happening over here? Basically, what is happening is that when the fat is solidifying, it is solidifying in the form of beta prime form, and over here this beta prime, they are getting transformed into more stable polymorph, which is regarding as the beta form, and hence there is a beta prime to beta transformation. So the beta prime, it is a high energy state, and beta is a low energy state. So when beta prime is converted to the beta state, that high energy state, from high energy to the low energy state, when these polymers are converting from the high energy to the low energy state, it is what you call liberating the extra energy which is there within its structure, and hence we are looking at this what you call these exothermic peaks. Okay. Now if you look over here, the rate of crystallization, if you look from SW1 to SW4, the rate of crystallization was decreasing as we were adding CCNTs. However, in SW5, if you see, there was a rapid increase. Okay, that is the reason. Due to the rapid increase, if you see, the crystals were not formed in a proper manner. Okay, so the same thing you can see over here. Since the average crystallite size, it is just 8.745. So you can see everything is falling in place now, and everything is being correlated with the microscopic structure, right? So whenever we do the research, we look into these parameters, how all these experimental parameters are related with our observational parameters, right? Again, then we did DSC. So in this DSC, this first peak is for the alpha polymorph. Okay, then we have this beta prime. This blue arrow is the beta prime, and this one is the beta polymorph. Okay, so this is the high energy state, so it takes lower energy to get molten beta prime, and beta polymorphs, they are the most stable thing. Okay, now when we are adding these CCNTs, the peak, alpha polymer peak, which was present at around 20 degrees centigrade, it appeared at a lower temperature, which corresponds to the gamma polymorph of the fats. Okay, and this gamma polymer was present in each of the formulations. And another interesting thing is that in all these formulations, we were having beta, a prominent beta transformations, and there were some shoulder peaks, and that is this is why, if you see, predominantly we are getting this beta polymorph from this system. Okay. Over here also we are getting that predominantly we are having the beta polymorph within the oleogel system. Then we deconvoluted these thermograms and we try to add up what is the area. So if you see, this is minus of 3.992. There was a reduction. Then there was an subtle increase over there. And if you see over here on this SW4, it was having an area of 3.983, and lastly SW5 it was having an area of 3.773. Now among all the CCNT containing systems, if you see this SW4, it was having the highest added area. That means this formulation is the most stable in terms of the thermodynamic or the thermal properties. Okay. Then we did rheological analysis. In the rheological analysis, we did the stress relaxation study. So if you see the SW3 and SW5, they were during the test, they were breaking down. Okay, they were not able to take down the stress. Now if you see this stress where the breakdown happens in SW5, it's quite early. Okay, so if you correlate with the micrograph, we were seeing bicontinuous phase, and it is the characteristics of the bicontinuous formulations that they do not have a very strong architecture. Okay, and if you see in SW4, it was having a very good this thing, even better than the original SW1, the piston oleogel. If you see, there were in very a large number of ripples over here. Now these ripples are due to the micro disruption within the 3D structure. Now if you look over here in SW4, it is quite smooth. So this SW formulation, it is quite a stable formulation even in terms of the mechanical properties. Okay, so we found out the percent stress relaxation. If you see, the percent stress relaxation was the highest in terms of in SW5, followed by SW3. So why these are so? Because if you see, there was a structural breakdown, right? And due to that, the relaxation has happened quite quickly. Then we did the drug release study, and if you see, as the CNT concentration was increased, there was a decrease in the CPDR percentage. And why this is so? This is because it is a reported fact that that CNTs, they have a channel. Okay, they have a channel-like structure. So usually the drug diffuse within this channel, and since there is a very high surface to area ratio, so the CCNTs, they absorb a large amount of drug molecules within its structure, and that results in the controlled release of the drugs from the CNT containing systems. And this is what we are observing over here. This is purely basic. This observation is purely due to the CCNTs. Okay. Now then we fitted the model through coursework papers model and paper saline models. Okay. So these are the model fitting parameters. I'm not going into the details. However, in GIST, if you see the K parameters, which is the diffusion constant of the drug within the oleogels, SW1D, it was the highest. And as we go from SW1 to SW5D, the diffusion was, internal diffusion was reduced. And that can explain the reduced drug release from the oleogel systems. Okay. So paper saline model, it is a more complex model as that of the correspondence papers model. It is a generalized model where you have diffusion due to the Ficant constant or Ficant diffusion, and there is another parameter which is called the diffusion due to the relaxation of the network. So there are these two types of parameters which are there. Okay, so I'm not going into the detail. I think I have already consumed my most of the time. So in GIST, what we have done, we have prepared shea butter and wheat germ oil oleogel. So this was prepared. Then we added CCNTs within these oleogels to prepare a series of oleogels, and we saw the changes in the key properties. We saw that there are changes in the rheology, there are changes in the XRD, also there are changes in the drug release pattern. So I think I have already discussed most of the things. The main thing is that the main outcome from here is that the CCNTs, they have been used, we have used it as a successful crystal modifier to alter the morphology of the globular crystals of uh, this shea butter. Okay, so I'm just going to the last slide, last point. So here in this study, we can conclude that CCNTs can potentially be used as the crystal modifier of fat networks. Okay. So I would like to thank my collaborators. So these are my collaborators from India, and I have my collaborators from the international collaborators. I believe Professor Slawomir is also here, and tomorrow he's giving a lecture, or maybe today itself he's giving a lecture just after me. I thank him for his continued support towards whatever research he is doing, and we are collaborating with him. Okay, thank you, Professor Slawomir. So I would like to thank the different funding agencies. So I would like to thank the Department of Biotechnology, then Department of Science and Technology and Science and Engineering Research Board from the Government of India. I would also like to thank Science and Technology Department of Government of Odisha for helping us with funds in different for different aspects. Okay, so you will find a lot of work what we do. We put those up in our YouTube channel. Please do not forget to visit our YouTube channel. You will find a lot of research resources for doing your research. And if you need any help in doing any research, you can contact me by email. Okay, so thank you all. So at last, I would like to say that teachers thrive on the success of their students. So I would like to thank my students who have been working hard, and because of that reason, I have been able to what you call discuss whatever the research are going on in our lab with you guys. Thank you all. Thank you for your patient hearing.
Thank you, sir, for your nice and excellent presentation. And we have know about your work, and basically your work is your oleogel related product. And before the drug release inside the body, we should know the different physiochemical property. So during analysis of the work of your, we can get that SW4 is the most stable product. So in this manner, our participant also get the knowledge from your work. So, as you know that our theme is new horizon in pharmaceutical research. So many of the participants didn't know the about this oleogel related product. So I must sure that many participants of this area can get the knowledge of the basically it is a pharmaceutics related area, and some phytochemical also there. So many participants can know this new research area, and they can explore in this area coming years. And one thing I also congratulate you that your work is also get presence all over the world. So I am must congratulate your work also. So thank you, sir, for your nice presentation. And again, I thank you given us time for this conference with your busy schedule. So thank you, sir.
Thank you, Professor Dalai.
So over to
Thank you for giving me the opportunity to present over here.
oh, thank you, sir. So, so we have the next session. So we have start with a new resource person. So I over to Subhasri Madam to continue the proceeding.
Thank you very much, Professor Dr. Kunal Pal, sir, for a beautiful and valuable presentation, and thanks for taking time out of your busy schedule. Actually, I like to thank personally, sir, your encouragement for this international conference matters a lot. The discussion about oleogels and incorporation of bioactive agents like curcumin, vegetable fats, natural products in oleogels are very useful informations for ongoing research. Incorporation of mango water from mango seeds and shea butter in pharmaceutical dosage forms and the pattern of data interpretation and analysis and different analytical techniques which you have followed during this experimental design is really appreciating, and definitely we will follow these ideas and our participants will follow all these ideas. And the discussion about solid lipid nanoparticles is also valuable for us. Now I request our chairman sir, Dr. Biswaranjan Patnaik sir, to felicitate Professor Dr. Kunal Pal sir.
नमस्ते डॉक्टर पाल।
नमस्कार सर, लास्ट टाइम आपसे मुलाकात नहीं हुआ, सॉरी फॉर दैट।
I was hearing about your product, oleogel and shea butter. It's very nice. And
Yeah, thank you very much.
I'm very much interested for the developed product and to develop it further to the pilot project and to the commercial use of it. So we'll see how far it will be possible to develop in the pilot project and go for the commercialization.
Yeah, sure.
I have also developed 2-3 products, commercial product, but not this in this line. That is inorganic electrolytic chromium. That is pure zinc, ash. So anyway, we'll discuss further. So I am requested to honor you by giving our certificate. I hope we'll accept it as token of honor to you. Professor Kunal Palji, honor given by the KANAK MANJARI Institute of Pharmaceutical Sciences.
Yeah, thank you.
And we like to see you personally.
Yeah, sure. It would be my pleasure to meet you.
Thank you very much. Thank you for your delivery.
Okay, thank you. Namaste.
Master, बहुत तो दरकार नहीं किसी.
Now, already we have completed one beautiful session by Professor Dr. Kunal Pal sir. Now, I am going to welcome Professor Dr. Slawomir Wilczynski as the head of the Department of Basic Biomedical Sciences, Medical University of Silesia, Poland, as the second keynote speaker of this session. He has been working in the field of cosmetology and dermatology. He pursued his Masters of Pharmacy in the year 2003, bachelor's degree in cosmetology in the year 2005 and doctoral of pharmacy in 2008. He also had pursued Masters of Cosmetology in 2017. He had a good research experience in skin diagnosis with a particular emphasis on image analysis and processing methods, application of hyperspectral and thermal imaging in cosmetology and dermatology, and so on. He receives many research awards and honors from different universities for his eminent research work in the field. He has so far authored more than 95 publications with impact factor 101 and 3 patents. He is also an editor-in-chief of the magazine in the field of cosmetology and aesthetic medicine, Aesthetica, and he is also the member in scientific societies. The article profiles a number of people who are making a difference in the world, and you can find him at the top of the list. For this immense knowledge in the field of research, his dynamic range of acquaintances, and a sense of adventure that knows few bounds. I would like to introduce, on behalf of KANAK MANJARI Institute of Pharmaceutical Sciences, Dr. Slawomir Wilczynski. Sir, welcome, please.
Thank you very much for your nice presentation. I would like to thank Professor Shari Sahu from KANAK MANJARI Institute of Pharmaceutical Sciences for the invitation. It's an honor for me to be able to share the results of our research with you. And I would like also to thank all my Indian friends, and especially I would like to thank for Professor Kunal Pal of the National University of Rourkela, Technology Rourkela. So thank you very much, and I'm going to present you some research concerning, I'm sorry, just a minute, I'm trying to present my presentation. Just a second.
Slawomir, sir. Hello, sir.
I'm sorry, I have some technical problems with presentation. So give me 2 minutes, please, okay?
Thank you, sir.
Okay, so can you see my presentation?
Yes, sir. Yes, sir.
Okay, thank you very much. So I'm sorry for the technical problems. I would like to say you something about the new methods of identifying of the counterfeit drugs, and so why this problem is so important. When you see what's the best business in the world, so let's say you invest $1,000. So if you invest $1,000 for counterfeiting money, you can earn $3,000. In credit card falsification, you can have $6,000. For prostitution, 10,000. Cocaine, I mean, not producing but smuggling cocaine, you can earn $20,000. Counterfeiting cigarettes, $40,000. So if you invest just $1,000 for drug falsification, you can earn from $200,000 to even $500,000. So it's one of the best business in the world. So that's why drug counterfeiting is so big problem for modern medicine. And according to WHO statistic and FDA statistics, it's estimated that about 10% of the global drug market is the drug counterfeited drugs. So about $35 billion in 2005. This market was worth 75 billion in 2010, and even $120 billion in 2020. So so we can see a big increase of prevalence of this thing. So how many drugs is counterfeited? It's about 1% in developed countries, since even 30, even 50% in some African countries. So in 1914, 116 countries identified this problem in their market. So the drug falsification is the very big problem, but it's not very popular. Not many publications are concerned that this problem, and if we wonder how many people dies because of drug falsification, the statistics are really bad. So more than if we sum up the deaths caused by HIV virus and health problems, more people dies due to using falsified drugs. So this is really big problem, and so we are trying to find new methods of falsified drugs identification. And as we see on the statistics, every 5 years, the number of disclosed cases of counterfeit drugs is doubled. So this procedure is more and more popular across the world, and so we are trying to find out how to fight with this drug falsification. There are many manufacturers of the falsified drugs, especially in China. And then these drugs are exported over the world. And how does this look in statistics? According to WHO statistics, in counterfeited drugs, about 30% of the drugs do not have any active pharmaceutical substances. About 20% of counterfeited drugs have incorrect amount of active pharmaceutical substance. Product with incorrect active substance is about 20%. Product in incorrect packaging is about 16%, and copies of the original product, copies I mean the active pharmaceutical substance and other substances are identical than in original product, and the copies of the original product is about only 1%, and products with a lot of impurities is about 9%. So which one of this product is more dangerous? Of course, the first one. So the products without active substance are the most dangerous, because let's try to imagine that we are trying to heal the cancer, and we or something bacterial, and we are healing this using drug without active substance. So this is one of the most deadly counterfeit products. And due to complexity of the pharmaceutical market, especially in Europe, it's quite easy to add some counterfeit products to legal sellers. So I mean hospital, patients, especially pharmacy, wholesalers, and we can find counterfeit drugs in all countries across the world. Of course, in Poland also it's a big problem. It's very difficult to say how many drugs are falsified, because the problem of drug counterfeiting and drug falsification requires a lot of money, because trying to find and analysis of a lot of drugs is very, very expensive. So nobody knows what's the percentage of the counterfeit drugs in the illegal market across the world. As I said on the beginning, WHO states that about 10% of drugs on global market are counterfeited. And why this is so dangerous? As I said, it's dangerous because for the patient, because a lot of people die for this reason, but also other purposes. So economic loss, wasted resources, increased out-of-pocket spending, economic impact. In case of socio-economic impact, lost productivity, lost income, lack of social mobility, and of course the most important, the health impact, higher disease prevalence, progression of antimicrobial resistance, a loss of confidence, increased mortality and morbidity as an adverse effect of using counterfeit drugs. As I said, there are hundreds of thousands of people, maybe even millions, because even the WHO does not have the statistic how many people dies because of using counterfeit drugs. We don't know how many people dies because we don't even know how many drugs are falsified. And the penalization. Penalization is not very strict for drug falsification. So according to European law, who produces a counterfeit drugs, a product, or falsified active substances shall be subject to a fine, imprisonment for up to 5 years. If you compare it, for example, cocaine or some other things, it's not so much. So as I said on the beginning, you can earn a lot of money. The risk you have been captured is very, very low. And if you are captured, the penalization is not very strict. So that's why the problem of drug counterfeiting is so increasing. And how does the manufacture of counterfeit products? Of course, this picture is not it's not from the fabrics, let's say fabrics or manufacturers of the counterfeit products. These are pictures from manufacturer of counterfeit products. As you see, it's difficult to say even of counterfeit of manufacturing. It's It's unbelievable that people are using these products. Some of these pictures comes from Poland, because Poland is also a big manufacturer of counterfeit products. So as you can see, very primitive technology to counterfeit products. They do not the drugs which are produced in such a way are very, very dangerous. And how to export counterfeit drugs to other countries? The most popular is by small packages. If you send a small package and the value of the package is less than $10, no one and never will check what's inside. So it's a very easy way how to export some counterfeit drugs to other country. I would like to say something about very interesting phenomena. I mean heparin crisis. As you see on the picture, heparin and chondroitin are very similar. The structure of heparin and chondroitin are very similar. Of course, the action of these two substances are quite different, and heparin is one of the most important drug. And how do the heparin is produces? It's produces by long bowling of a pig's gut, and that's the way of producing of heparin. And in 2008, there was one of the biggest crisis in the pharmaceutical market, and the name of the crisis was heparin crisis. What and how happened? Only 4 manufacturer produces the heparin. All these manufacturers were in China, and this manufacturer produces heparin, and the price of heparin was about $900 for a pound of heparin. So they saw like this: if we add some chondroitin to heparin, because they are very similar, they have very similar chemical composition, maybe no one will find out and we can earn more. So they add some chondroitin to heparin, and they sell it to pharmaceutical companies, big European, American pharmaceutical companies. And due to these substances are very similar, nobody find out that there are some chondroitin in heparin. And so they added more and more and more chondroitin to heparin, and there was and what happened? The heparin was not efficient. So a lot of people around the world started to dying because heparin was not effective. So there was one of the biggest crises in the pharmaceutical market. What is the most commonly falsified drug in Europe? Of course, it's type 5 phosphodiesterase inhibitors, and the name of drug is Viagra. Is the one of the most common falsified drugs. And when you see how many sildenafil on the tablet contains counterfeit products labeled Viagra, you can see that it can be from 0 even more than 200 milligrams. So if you take a tablet with more than 200 milligrams of sildenafil, it's very, very dangerous, and a lot of people dies of because of this using of these falsified drugs. And what is the number of buyers of PDA inhibitors in legal and illegal distribution in selected 3 European Union countries? Let's say in Europe, in Germany, about 27% is from illegal distribution. In France, it's about 15%, and in Great Britain, it's about 48%. So, as you see, a lot of drugs comes from illegal market, even in Germany, France, Great Britain. So I suppose in Poland it will be much higher, because we are not so developed country as Germany, France, or Great Britain. So I said about drugs, but the problem of falsification is also important for dietary supplements. And what are the statistics for Poland? So let's say about 6% of dietary supplements do not contain any active substance. About 34 active percent active substance is accordance with the declared other active substances, and about 31% active substances is not accordance with the declared. So as you see, more than less than 10% of dietary supplements have the active substance and excipients in accordance with the declared. Okay, so let's say from the few samples, Sidra is a Polish supplementary dietary supplement, and it's labeled that it's a herbal dietary supplement for erectile dysfunction. But as you can see, it contains also sildenafil, so it's of course counterfeited. The same situation for the dietary supplement Satibo, determined by CMS. Present, it contains sildenafil and tadalafil, so it's extremely dangerous because it's labeled as the natural product, contains only extracts from plants, but it contains quite big amount of sildenafil and tadalafil. The same situation with next product. It's labeled at 100 natural and herbal, but contains a lot of sildenafil in each capsules. Not only drugs for people are counterfeiting, but only for animals. So a lot of animal drugs are counterfeited, and also in this case the number of falsification increase, and every 5 years the number of falsified animal drugs is doubled. So how can we identify counterfeit drugs? Okay, so there are many techniques are used to identifying counterfeit medicines, and in particular chemical analysis. These methods are expected to have high sensitivity and resolution, and today chromatographic methods are mainly used. I mean, thin layer chromatography, high performance liquid chromatography, and also spectroscopic method. I mean, mass spectrometry and its derivatives. So I mean, liquid chromatography, mass spectrometry, and gas chromatography, mass spectrometry, as well as Fourier transform, infrared spectroscopy, Raman spectroscopy, nuclear magnetic resonance spectroscopy, X-ray diffraction spectroscopy, and This chemical analysis method evaluate the chemical composition of the drug and compare it with the reference, the original drug. Due to very high sensitivity of this method, even small difference in the chemical composition are detected and may be a parameter identifying the genuine or falsified drugs. Nevertheless, according to WHO, even up to 10% on counterfeit drugs can be expect copies of the original medicines. So they have exactly the same quantitative and qualitative composition in a term of both active pharmaceutical ingredient and excipients. Thus, the identification of such a drug using chemical analysis method can be very difficult or even impossible. And these chemical methods are very time-consuming and very expensive. So you have to study all tablets one by one, and identification one tablets it can take even a few hours or more. So if you have, let's say, 10, 100 on 1,000 tablets, it's not possible to find out which is original and which is falsified. So we are trying to find a new methods which would be as effective as chemical methods, but cheaper, much cheaper, and also faster than chemical method analysis. And therefore, we focused on image analysis and processing methods, which are, we think, which are very good for identification on counterfeit drugs. And first of all, I would like to say something about hyperspectral imaging and the use of this method for the counterfeit drugs identification. And the hyperspectral imaging allows to the spectral analysis of each pixel of image separately, enabling, among others, analysis of the homogeneity of ingredients distribution, which may be additional diagnostic criterion in distinguishing between genuine and counterfeit drugs. At the same time, the use of image analysis of processing method, including the applied methods of, for example, as I see further, principal component analysis, can be effective, enables effective visualization of the results. And in turn, molecular hyperspectral imaging is a technique in which each pixel that includes the complete spectral cross-section, I mean spectral signature of whole spectral range, is additionally subjected to analysis. This provides not only the image of the object, let's say a tablet, in the wide discrete spectral range, but also spectral information that allows the quantification or quantitative analysis of the active pharmaceutical ingredient and excipients. So let's say more. So for in our studies, we used KANAK hyperspectral scalar specking. It's from Finland, and we proposed a new method of image analysis of this result using GLCM algorithm. It's a gray level concurrence matrix. This algorithm enables to identifying the contrast in the image. So the contrast in the image is very specific for the drug, and that's why we can identify the counterfeit or genuine drug with very high sensitivity. Even this sensitivity is about 100%. To visualize the results, we can use, for example, principal component analysis, which enable us to separate the differences in the wavelengths for hyperspectral imaging. So if you see on the reflectance difference for the original drug and counterfeit drug, you can see that is a big difference in reflectance, and the biggest differences is measured for 690 nanometers, and on the picture on the right you can see the tablets of the Viagra. Half of the tablet is counterfeited, half is original, and you can see the visualization of the reflectance differences on the surface of the tablet, and you can see even on the visual way you can compare the drugs and say which one is counterfeited and which one is original. So the hyperspectral imaging is a very effective way to distinguishing between original and counterfeiting drug, nearly 100 probability between original and counterfeit. What is important? This is not destructive technique, and allows to perform a screening test. A dozen or even a dozen of samples can be examined at the same time. So this is quite reliable, time-saving, non-destructive analysis with for a quick withdrawal counterfeit drugs from the market. And on the picture you can see visualization of original and counterfeit drug. The second method, which is I think is very interesting, is the directional reflectance analysis. Why it's interesting? Directional reflectance analysis is mostly used in space industry, but I, with my team, use this method for counterfeit drug identification. So directional reflectance analysis, and we can by directional reflectance analysis we can compare the reflectance in a specific spectral range from the original and counterfeit drugs. In this case, we use commercially available directional hemispherical reflectometer, Survey Optics Corporation, San Diego, and this measurement had the integrated surface reflectance of surface of two different angles of its incidence, 20 and 60 degrees for 6 discrete wavelengths. So using this technique, we can compare the directional reflectance for the counterfeit and original drugs. And what are advantages of this method? First of all, it's very fast. So the analysis of the single tablet is about 2 seconds. So in 2 seconds, we have complete analysis, and we know if it's original of it or it's counterfeited. And it's also very simple because the measurement is very easy. Look, how does the reflectometer looks like? So you just put the trigger, and after 2 seconds you have the result, and that's all. So it's really simple, and it's not so it's not very expensive. It's much more cheaper than chemical analysis. I mean, for example, let's say mass spectrometry or other spectroscopic methods. And the next method I would like to say you something is a dynamical thermal analysis. This method is not very precise, but it has its own advantages, because we used a very simple thermal camera, and we measured the time constants of time constants, heating time constants for counterfeited and original drugs. Original and counterfeited drugs were heated up to 60 Celsius degrees. That's the temperature which is not harmful for the tablet. And then we were trying to find out what's the dynamics of the thermal changing for counterfeited and original drug. So the use of dynamic thermal analysis supported by image analysis and processing method enables to distinguish effectively between genuine counterfeit drugs and why it's so effective. Because when we determination of the time constant is particularly important because this parameter is independent of such critical factor as the object emissivity, variation, excitation amplitude, environmental factors, and so on and so on. So we can automatically detect of counterfeit drugs with the comparison of methods of tablets production by assessment of thermokinetic parameters are different for counterfeited and original drugs, and even changes in drug structure in case of the same composition can be seen by the difference of thermokinetic parameters. So this method is not so fast as reflectance analysis, but is very good for this is very effective in case of trying to find which one is a counterfeit tablet using a big amount of tablets. So this use of dynamic thermal analysis is effective. It's cheap, and or I would say it's very cheap and fast method. And do not require expensive equipment or special sample preparation, and so that's why I think it's quite interesting method. On the picture, you can see the original and counterfeited Viagra. A, it's original, and B, it's a counterfeited. They are quite similar. If you are not pharmacist or specialist, in the visual way, you are not able to say if it's original or it's counterfeited. And on the thermograms on the right, you can see a big difference in the thermokinetics parameters of these two drugs. So on the visual way, there is no difference, but in the thermokinetics parameters, the difference is very big. So the thermal profiles for counterfeited and original drugs, when you compare this thermal profile, you can very sufficient and very effective compare and identify counterfeit drugs. And one of the last methods I would like to say something is the NMR field cycling technology. H1 spin lattice relaxation studies have been performed in order to differentiate between original and counterfeit product, I mean, in this case, Viagra. The relaxation experiments have been carried out in frequency range from 4 kilohertz up to 40 megahertz, and in case of counterfeited product, the relaxation process is B exponential, have two components in whole frequency range, while for the original Viagra, the relaxation is always single exponential. Already the qualitative observation allows to distinguish between the original and counterfeit products. Moreover, a quantitative analysis of relaxation dispersion profiles have been performed in order to reveal the motional processes contributing the relaxation and determine the characteristic parameters. It has been also shown that on this picture that vibrational spectra do not show sufficient difference to be used for identification of a counterfeit product. It also was demonstrated that original and counterfeit products cannot be distinguished by means of structural studies, even very advanced ones. So on the picture you can see atomic force microscopy for counterfeited and original Viagra, and these images are very similar, so you cannot differentiate from the counterfeit or original one using atomic force microscopy. And the last technique I would like to focus is X-ray microtomography. In this case, you can see 8 samples of falsificate Viagra, and with the T is marked the original one. So when you see the tablets in microtomography visualization, you can see a big difference in this case. So when we use image analysis and processing method on these X-ray microtomographs, we can find out which one is counterfeited and which one the drug is original. So we used a lot of image analysis method, for example, GLCM, quadruple decomposition, and we was trying to find out if brightness, contrast, and homogeneity is a good way to compare the counterfeit and original drug. And what we can see, we can compare and we can identify a falsificate drug with 100% of specificity using this method. So this method is not cheap, it's quite expensive because microtomography is expensive equipment. It's also very time consuming because a single measurement takes more than 4 or 5 hours, but it's non-destructive, this is advantage, and it's very, very sensitive. So to sum up, we have few different methods we can adopt to identification of drug counterfeiting, but I think one of the most interesting is using directional reflectance analysis. It's cheap, it's reliable, it's very fast, and you can compare any drug. I mean, not liquids, but semi-liquids and tablets. You can for the tablets you can use the directional reflectance analysis for identification of falsified drugs. So I hope I focused your attention on the problem of drug falsification, because it's one of the most problematic thing in modern medicine. Thank you very much for your attention. Do you have any question?
Any question?
When you will come to India?
Slawomir sir, our chairman sir is asking you when you are coming to India.
Thank you very much.
I think no question from the participants. Thank you very much, sir, for your nice presentation regarding counterfeit drugs, identification of counterfeit drugs, particularly spectral analysis, directional spectral analysis of counterfeit drugs, and different techniques how to detect these counterfeit drugs. Really, these things are very valuable for us, and definitely these are very new things. Identification techniques which you have interpreted are very novel and very beautiful things, and in which way you have narrated and explained is also the matter of appreciation. Now I request our Honorable Chairman Sir, Professor Dr. Biswaranjan Patnaik Sir, to felicitate to Professor Dr. Slawomir Wilczynski.
Thank you, Dr. Wilczynski, for your talk about the identification of counterfeit drugs. Very nice talk. Now we like to show our respect and honor to our certificate declared by the committee. Please accept it.
Thank you very much one more time. It was honor to present our results for you. Thank you very much to everyone, and it was really, really great pleasure for me. Thank you very much.
Please accept it. This is the certificate of honor. So let us see in the future. Meet again.
Thank you very much. I hope we can see not online, but we can maybe you can visit Poland. You are great welcome, and I enjoy to and I would like to invite you for cooperation. So thank you very much.
Okay.
Excuse me, sir. Hello.
Yeah.
Sir, I am a participant who just got your valuable views on counterfeit drugs, and I'm really very much excited to know about our studies for the first time. For the first time of a foreign teacher, and and this is the first time for me to attend an international conference on this pharmaceutical research topic, and I'm really very much impressed through your through your reviews on that counterfeit drugs topic, and thank you, sir.
Thank you very much. It was great pleasure.
Yeah.
Thank you, sir. Now I request Professor Dr. Manoj Kumar Dalai, Department of Pharmaceutical Chemistry, KANAK MANJARI Institute of Pharmaceutical Sciences, Rourkela, to handle the session. Over to Professor Dalai, sir.
Thank you, madam, for starting the session. So now, good evening, participants, and we will start.
Sir, please switch on the audio and video. Audio is on, but video.
So now we will start presentation of participants. So first, the person of the presentation, Priyabrata Panda. I like to invite Priyabrata Panda to show the presentation. So participants have 3 minutes. Within 3 minutes, you complete your all the formalities. This in question and answer.
Yeah, hello.
So now start
Hi,
the
good evening, everyone.
Priyabrata Panda, your slide.
Yeah, sure. I am presenting my slide. Uh, One second, sir. You enter screen. Uh,
So before starting, next participant Pradhan, Abhishek Pradhan is there. Abhishek Pradhan. If you there, then you present. Next, you will show your presentation after Priyabrata Panda. So now you start your presentation of Priyabrata Panda.
yes, sir. Sir, is my screen visible? Hello.
Now visible.
Is my screen visible? Yeah.
Ah.
Okay. Okay. A very good evening to all of you. My name is Priyabrat Panda, and I'm presenting my an oral presentation on the topic of hybrid clinical trials versus virtual clinical trials. What is the future? So thanks for, and I very much appreciate that I am participating in the International E-Conference 2021 New Horizons in Pharmaceutical Research. My topic is on clinical trials. So recently, hybrid clinical trials and virtual clinical trials are new in the market. Means not new. It's like a topic of discussion in the market after COVID-19. So I would like to present my topic on this. My content for this particular seminar, it will be on introduction to decentralization in clinical trials, and then what is virtual trial and what is hybrid trials, and then what is the comparison between the hybrid trials, virtual trials, and traditional clinical trials, and which one should be the future as per my opinion. So before coming to so let's start with introduction to decentralized clinical trials. Coming to decentralized clinical trials, why decentralization clinical trials is the topic. To understand it, we have to understand that the general overview of a clinical trial. The general overview of a clinical trial, when we see, as you can see, the first step is the new drug discovery by a pharmaceutical company, proceeded with a successful preclinical study, and then filing for IND, investigational new drug filing. Then coming to the phase 1, 2, and 3 clinical trials of a particular drug by the volunteers visiting the site. This is very much important. Volunteers visiting the site, and after taking their consent, the 3-phase clinical trial is being completed, and the NDA is filed. After that, the drug is available in the market for human consumption, and post-marketing surveillance begins. Now, the problem with the traditional clinical trials is subject retention. Clinical trials are can be very much become successful if the subject are participate in large number. But if subject participate in a very less number, then the success rate of a clinical trial declines. So most trials experience the issue of missing data, often referred to as loss to follow-over, attrition, or dropout. And this can bias the findings of a trial. So the factors that improves, that promotes the subject retention are the age of the subject. If the subject is a geriatric population or pediatric population, then there may be less recruitment. The gender, women volunteers, female volunteers versus male volunteers, reluctant to visit the study site multiple times. The third point actually is very much important, because generally when it comes to clinical trials, it doesn't happen very quickly. The particular subject has to visit the study site, has to stand in the line, has to fill the forms, has to undergo training of ICF, then only he can be included in the clinical trial. And after that, there will be multiple visits. So there is very much chance that the particular subject may get frustrated due to regular visit to the particular clinical site, and then there may not be a touch up with the volunteer. Similarly, mental health, study duration or length, and recently
So Ponda, I
COVID-19. These are
interrupt
also very
you.
much.
Mr. Ponda, I interrupt you.
Yes,
Your
hello.
slide is not showing completely. You go one by one.
Not showing completely.
Yeah. Only first slide is
Is it
showing.
okay?
You go one by one.
One
It's
second.
very quickly.
No, sharing another application. Is it? I am actually going with
No,
the
no,
first slide.
actually, I would like to interrupt Priyabhata Panda. When you are presenting, you have to, in PowerPoint, you have to share the entire screen. Then only your presentation will be visible.
Okay.
Share your entire screen.
Okay. Okay. Just one second. I will again present. Entire screen. Okay, one second. Screen. 1 second. Is it visible now? Hello.
Now visible. Now you go
Yeah. Is it visible now?
F5.
Okay.
F5 for complete screening.
Yeah. Yeah. F5.
F5
Yeah, it's there. F5. Yeah,
or in the bottom tab,
Yes.
you just click the cup. You just click the cup. In the cup symbol is there.
Yeah, I have present.
Or switch
what's
over
showing?
to F5. F5.
Yes, sir. I have switched on to F5 only. It's completely, my entire screen is with my PPT only.
But
But
we are
Is it
not
visible now? Introduction to
cover the
decentralized.
visible, not covering the whole window. It is only covering the.
okay.
Okay. 1 second, sir. 1 second. Let me again present your entire screen. 1 second, sir. Select window or screen.
Your monitor is also showing full screen, no? Whenever you are presenting any PPT, this will cover full screen, so that it will show prominently to
Okay,
the
yes.
participant.
Okay. 1 second, sir. 1 second. Is it visible now?
Not visible.
Is it visible? 1 second, sir.
So every participant, they will practice how to display the full screen before presenting. So
Sir, is it
during
now
this,
visible?
it is visible, but not full screen. Anyway, you just present now.
1 second, sir. Why it is not presented?
So you will take more time. So you go one by one through the. You are not incapable to in full screen.
Let it be like this. You just change the slide One by one.
You go.
Okay. Okay. So, okay, fine. So I will be carrying out with this since it's getting little. I'm not
Now
getting the
you
point
go
how to
second
get
slide.
full screen.
Second slide. Already you're there. We go quickly go second slide.
Yeah, second slide is there, right? Introduction to contents. Content section is there, right? Yeah. Um, is it okay now? Hello.
Okay, but you are still in first slide.
Um,
You just move on with the arrow mark.
yes, sir. I'm going that way only. Okay,
You
so
move your cursor to second slide,
You click the
or
second.
you go to
yes, sir. I've
the
done. I'm in the
arrow tab.
slide. Yes, sir. I'm in the. Okay, okay, fine.
Okay, okay.
Okay, fine.
Now,
I will go like this
now,
way, sir. Okay.
uh,
Okay, fine.
good.
So let me zoom it. Let me little bit zoom it so it will be okay. Um, so as I said in the starting beginning that when we go for a decentralized trial, the concept of decentralized trial, before understanding it, we have to understand that how a traditional clinical trial works. So the new drug discovery must be done by pharmaceutical company, then the preclinical trial, and then voluntary visit, and then the drug is available in the market. This is a traditional setup. Now, in that particular setup, the problem is with the subject retention. That subject don't come to the clinical trial oftenly because it involves many factors like the age of the subject or gender or reluctant to visit the site multiple times, mental health, trial participants, compatibility between key aspects of the trial, and recently COVID-19. So when subjects don't participate or like there is no retention of subject, this leads to longer duration of the clinical trial, increasing cost of the clinical trial, and then less statistical power, termination of the trial in some cases, and prevent greater efficiency in clinical development. So patient-centric clinical trials are the need of the hour. So with that particular concept in mind, the decentralized trials has come as a solution to make clinical trials more patient-centric. So earlier, when the new drug discovery was being done by pharmaceutical company, now also the new drug discovery is being done by pharmaceutical company. Successful preclinical studies are being done. But in the third stage, where the particular subjects has to visit the study site, but now they don't need to go to the study site. That particular trial can be done from their home. They study site visit sites are not required. So remote patient engagement, direct to patient shipment, patient based apps, home nursing are some of the virtual clinical trials, or we may say pragmatic study based clinical trials, which can be done to help or to make clinical trials more patient centric. So there the decentralized trials concept comes, that a particular clinical trial or a particular subject doesn't need to visit to the site more often to get a clinical trial done. He or she can be there, be sitting at his home, and the trial can be conducted. So that is what the decentralized clinical trials concept is being now followed. So then why hybrid trial is necessary? Can't we go completely virtual? Now if you are a sponsor, what will you prefer? So whether you will prefer virtual or hybrid. Now we will understand that what is virtual trial and what is hybrid trial after understanding decentralized trial. So virtual clinical trials, as we say, virtual clinical trials are the new method of collecting safety and efficacy data from clinical trial participants, from study startup through execution to follow up. That means particular volunteers need not go to study site. It can be done from their home through virtual technology. Virtual clinical trials rely heavily on new technology, using anything from an app on a smartphone to wearable gadgets, collecting important health measurements to conduct each stage with comfort of the patients. As you can see here, telehealth is there, wristbands are there, gadgets are there, EICF, e protocols. These can be done so that patient can carry out clinical trials from their home. So then I will then, so how can you, so how patients will come to know that a virtual clinical trial is being held? What are the methods that,
So I
or what are
interrupt you,
the
Ponda.
routes?
Hello,
Yes,
hello.
yes,
Ponda, you have
yes.
only 1 minute left. So within 1 minute, you complete your all thing.
Okay, okay. Thank you, sir. So there are many apps present like Google Health Studies app, and then you can check out on clinicaltrials.gov, where you will find different virtual clinical trials going on at present or recruiting. Then there are WHO, ICTRP, International Clinical Trials Registry platform, and then there will be like Facebook. From Facebook also, volunteers are being requested to join clinical trials. And apart from that, different pharmaceutical companies also have their own particular app for volunteer recruitment. Like Novartis has their Focal View app, GSK have GSK Clinical Studies, and then Encapsia Home Visit app. So these are how a virtual clinical trial is being conducted, or they are trying to conduct virtual clinical trials through app-based systems. Now, the very first virtual clinical trial was being done by Pfizer, which was a remote. It was called as remote. It was on Detrol, tolteridone tartrate. It was a virtual clinical trial which did not succeed. Only 18 participants were participated in that particular trial. It had its reasons. Now, similarly, another virtual clinical trial example is insomnia-based trial, which you can find in clinicaltrial.gov, to test a medical device called as cranial electrotherapy stimulator device. It is also a virtual clinical trial where patient need to go to visit the site. So what are the advantages of virtual clinical trials? It enhances subject recruitment. It remote patient monitoring can be done, patient compliance trials and patient driven data collection, faster drug development decisions, more cost effective, and studies carried out from home. So after knowing all this, we can say that virtual clinical trial is the future, or virtual clinical trial is best. But wait, carrying out a clinical trial completely in virtual mode is still an exception. There are challenges to virtual study trial which can be fixed if a hybrid trial is brought into picture. So since my time constraint is there, I will just like to put on a fact that what is hybrid trial. Hybrid trial means traditional trials plus virtual clinical trials. When you mix them together, you get a hybrid trial. So let me give an overall picture of the differences. In traditional trials, there is randomization. In hybrid trials, there is randomization. But in virtual clinical trials, there is no randomization. Occasionally, there it is. Cost per participant is high in traditional trials. In pragmatic trials, it is lowest, and in hybrid trial, it is lower. And sample size depends on the size in all the three trials, but generally hybrid trials and virtual clinical trials have more patient based studies. So the advantages of hybrid concept trial is that patient data collected is of much broader spectrum, the cost per participant is low, presence of randomization and blinding, healthcare professional care level increases, lower retention rate, and decrease in number of sites needed. So during COVID-19, the CROs, Clinical Research Organization, can help sponsors in carrying out hybrid clinical trials because they have different technologies with them. So I think hybrid trial is the future of the new clinical trial era or clinical trial research. Thank you.
So Ponda, with this, stop your presentation. So you have your presentation showing many things. So it is not possible this time period. So anyhow, you manage the presentation. So thank you for your presentation. So one question from any participant, if you want to know. Any question from participant? Quickly put the question to the Panda so he can quickly able to answer.
Sir, please switch on your video, sir. Manoj, sir.
Hello? Now audible? Hello?
Sir, video.
Ah, So any question from participant? So my question is to Ponda that what are what are the advantage for if anybody start clinical trial from home base? You are saying that home based clinical trial. What is
Yeah.
your point of view?
My point of view is that home-based clinical trial completely, I don't think it's that much feasible, or like it's completely flexible home-based trial, because stool sample, urine sample, this can be conducted from home-based. But if there is a biopsy required in a particular clinical trial, then home-based study is not possible. The particular subject has to report to the clinical site. So that is the reason why hybrid clinical trials should be there. That some part of the study should be virtual, some part of the study should be site based, so that patient can enjoy both of these things. And that's in that way also the particular drug will have more chances to get succeeded in a particular clinical trial study, looking into the particular environment of COVID-19.
Okay, thank you. So with this next, I call upon next participant of this session. Next, Abhishek Pradhan. Can you show your presentation?
Yes, sir. Thank you,
Hmm.
sir. I show you a
So
presentation.
you have Abhishek, you have only 3 minutes. So within 3 minutes, you quickly go through your presentation.
Okay, sir. Yes, sir.
Please switch on your video, Abhishek.
Yes, ma'am. मैम मैम स्क्रीन शेयर में यूज नहीं हो रहा मैम योर वीडियो मैम हेलो मैम हेलो
Hello, huh?
Yes.
मैम स्क्रीन शेयर में वीडियो ऑन नहीं हो रहा सर
So anyway, show your
Okay, okay, you
presentation.
continue.
यस सर स्लाइड इज विजिबल सर
Ah.
विजिबल सर द स्क्रीन इज विजिबल मैम
Yes, yes. Yes.
ए वेरी गुड अप
No, your slide is not visible.
1 minute सर
So
Okay, now it is visible. Now it is visible. Carry on, carry on.
A very good evening to all, sir. I am Abhishek Pradhan, student of Century University of Technology and
Mr.
Management.
Pradhan,
Yes, sir.
now start your presentation.
Yes, sir. Yes, sir. Yes, sir.
You continue.
Yes, sir. A very good evening to all, sir. I am Abhishek Pradhan, student of Century University of Technology and Management, School of Pharmacy, Balangir campus. Today, I am going to present seminar on an advanced and innovative technique, osmotically controlled oral drug delivery system under the guidance of Mr. Chaitanya Prasad Meher, sir. First, we know the what is osmotically controlled oral drug delivery system, sir. This is the one of the most common promising drug delivery system. They're done by using osmotic pressure and its driving force for the controlled delivery of active agents, sir. And the main motive of the osmotically controlled oral drug delivery system is therapeutically effective and reduce the side effect of the drugs and viability of the drugs would be increased. Since here is my abstract, which clarify the conventional oral delivery system is little or no control over release of the drugs, and the rate and extent of absorption of drug from conventional formulation by very gratefully, sir. And the characteristic release of characteristic depend on the predictably easily property of the doses found, sir. Then come to the next slide. What is the OCDDS? Osmotically controlled drug delivery system deliver the drug in a large extent and delivery nature is depend upon the physiological factors, sir, in the gastrointestinal tract, sir. Then some basic factor affecting, sir, design of osmotically controlled drug delivery system like orifice size. Orifice size maintain the minimum and maximum value, sir. Method to create a delivery orifice in a thermostatic stable coating or mechanical drills or larger drills, sir. Then solubility, sir. Solubility depends on the solute inside the drug delivery system. Example, cyclodextrin enhance the drug stability or solubility. Then osmotic pressure. Osmotic pressure affects the rate of release. And to achieve the zero order kinetic in osmotic, then semi-permeable membrane comes to permeable to water and not permeable to iron, and release rate is essentially independent of the pH. And some basic components of osmotic system like semi-permeable membrane. In semi-permeable membrane should be inert and maintain its dimensional integrity. Examples, cellulose acetate of butyric, cellulose triacetate. Then osmotic agent. Osmotic agent maintain a concentration gradient across the membrane. Flux regulator, sir. Then flux regulator to regulate the permeability of fluid by incorporating the flux regulating agent like in the layer. Example, hydrophilic substances like polyethylene glycol. Then weakening agents, pore forming agent, coating solvent, plasticizers. Then there are some types of osmotic pumps like elementary osmotic pumps. This is the diagram of elementary osmotic pumps. In elementary osmotic pumps, a delivery of orifice part is above the diagram, and the middle part, outer membrane, is semi-permeable membrane, and the components, that is drugs plus osmogens components, are in elementary osmotic pump help to measure method of achieving controlled drug release system, sir. Then push-pull osmotic pressure. This is two parts. Push-pull, one is delivery orifice meter, sir, and black market rate sedimented is expanded pushes compartment during the operation, sir. And the one is osmotic drug core and the polymeric push compartment before operation, sir. Before operation and after operation, sir. In push-pull osmotic pump used for the very poor solubility and similar to the bilayer tablets, sir, using it. Then osmotic pump with non-expanding second chamber, sir, that used to dilute the drug solution which is released in the body. Then control porosity osmotic pumps that helps to control the porosity of osmotic pump. Tablet of diclofenac like diclofenac sodium tablets. Modify osmotic pump for insoluble drugs. Then modify osmotic pumps for highly soluble drugs. Then osmotic matrix system, sir. Then osmotic pumps for two or more drug. Then osmotic system for control delivery of liquid drug that help to active ingredient in liquid form are difficult, sir. Then mechanism. The mechanism of OCDDS is designed to a tablet core, including one or two osmosis agent with the drugs. Then tablets is lined semi-permeable membrane, so there is water flowing through GIT. Then passes the solution of preparation in zero order kinetics and rate determination your type of coating and formation of main compartments of the bronchial diameter, sir. And how then the OCODDS is evaluated by some points, that is categorization of doses from effective or effect of osmotic agents, swelling property, membrane stability, and thickness, orifice diameter, and drug release, and in vivo drug release study. Then it is in this osmotically drug delivery system clarified the or concluded that utilize the principle of osmotic pressure for the drug delivery system, and it is independent physiological factor of gastrointestinal tract, and it is help to decrease 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 the adverse effect or reduce the side effect, and viability of the drugs would be increased, and how to improve the stability or helpful or effective doses from each people in this dosage form. Thank you all.
So with this, thank you, Pradhan, for your presentation. So any question from participant, if you want to know their answer? So Pradhan, what type of instrument you are preparing? Abhishek?
Sir? Yes, sir.
What type of instrument your study will analyze?
Sir?
What type of instrument this require for your study? You are getting my point?
Yes, sir. Yes, sir. Sir, in this osmotically controlled
How
drug
you
delivery
measure
system,
osmotic?
in it is measured in pharma industry, sir.
Okay.
Pharmaceutical industry.
So thank you, Pradhan, for your presentation. So I name the next participant, Amitabh
you, sir.
Chatterjee.
Thank you, sir.
Amitabh Chatterjee, show your presentation right now.
Thank you, sir.
Namaskar and very good afternoon to the Honorable Chief Guest, Chairman Sir, and all the faculty members of KMIPS. I, Amidana Chatterjee, student of MPharm, today got an opportunity to give a poster presentation on a topic named as Pharmaceutical Industries in Nation Development. Basically, it's a survey being done by me by collaborating different articles that have been published on the newspapers as well as on the government websites. So basically now I'm sharing up the screen for the poster presentation. I hope so it is visible to all. Sir, is it visible?
Visual.
Okay. So firstly, as we all know, okay, sir.
Okay. You show your
So
study.
firstly, as we all know that India is one of the world's largest supplier of generic drugs. Our pharma sector has a supply of about 50% of various types of vaccines globally. If we talk about the recent contributions of the pharma sector for our nation development, then firstly, in November 2020, IIT Bombay has developed the products like alcohol-free sanitizers, portable sterilization devices for the hospitals, and so on. Then, in the year of 2020 itself, in the month of October, Aurobindo Pharma also acquired MV's Pharma Ventures by a sum of rupees 274.22 crores. And even in April 2020, PM BJP, that is Pradhan Mantri Bharatiya Jan Aushadhi Pariyojana, got a turnover of rupees 52 crores. Similarly, if we talk about the government initiatives towards the pharma sector, then our Indian government has unveiled Pharma Vision 2020 to make our country a leader in end-to-end drug manufacturing. And even according to the economical survey of 2020, our GDP percentage in the healthcare field has also increased from 1.2% to 1.6%. Also, in the graphical representation, we could also see that India tops the list of CAGR percentage, that is basically compounded annual growth rate percentage. And also, India is having a huge number of US FDA approved manufacturing plants. So not only in the field of manufacturing, but also in this pandemic period, we have proven our leadership among different countries by manufacturing the PPE kits within 2 months of COVID-19 outbreak. And now we are able to manufacture the PPE kits at a rate of about 4.5 lakhs daily. And even the vaccine which is being developed by our own Bharat Biotech, named as Covaxin, is even efficacious against the UK COVID-19 strain and also has no serious ill effects. Thus, I would finally say that our Indian pharma industries is helping our nation to build a strong relationship with other nations and also giving an economical boost to our country to grow globally. So here I would like to end my presentation, and basically I would specifically thank Subhashri ma'am for giving us an opportunity to be a part of this international seminar and share our thoughts, as well as we could also gain various amount of knowledge through different scholars of different countries. Thank you very much.
So thank you, Chatterjee, for your presentation, poster presentation. So now I am expecting question from our participant. If anybody interested to say any question to the Chatterjee.
Sir, Amit, can you
Yes,
say
sir.
what is the difference between this generic drug and branded drug?
So basically, generic drug, basically it is containing the API itself, and it is mainly being developed by the company which is mainly means having the right of patent actually. And after the patent gets over within a period of 20 years, then then other companies then other companies can take that formulation and can also start production of their drug, and in that case basically the generic drug becomes the branded drug, I guess, as per my knowledge, sir.
Okay. Thank you.
Welcome, sir.
So I have one question for Chatterjee. So you did a
Yes, sir.
survey among the pharmaceutical industry exists in India or outside of India also. You
Yes,
collect
basically, sir.
this is a
Yes,
collective
sir.
data.
Yes, sir.
So if you give some data value that how you make comparison, how you get a about summary of this survey. You have
Sorry,
not
sir,
showing
can
any
you
data value.
sir, I showed actually
Hello.
the graphical representation, sir. Yes, sir.
What is the data value you like to show? Mathematical value you are showing, no? So you have
So
make a survey.
so particularly as per the data value, it has been shown in that graphical representation that India is having more than means in that data actually it has been written that among different other countries for US FDA drug, the India is having the maximum amount of manufacturing plants. It is actually written there more than 100, but I guess that it is the value exactly has not been shown there. So that much I was
Okay.
knowing, sir.
Okay, thank you for your poster presentation.
Thank you, sir.
Now I like to call upon next participant, Aishwarya Nayak. Can you show your presentation?
Yes, sir. Yes, sir.
Now you go to the present now. If you are in Google Meet, directly go to the present now for your poster presentation.
So is it visible?
Ah, your face is visible.
So presentation?
Now show your poster presentation.
So is it visible?
No, no. Your display of poster is not visible.
May I interject? First of all, what you do, you open your PDF file or PPT file, okay, then present now.
So is it visible now?
No, no. It is not visible.
No, sir.
You are showing the Google Meet window. Now
Yes, yes, it is now visible.
Okay. So good evening, respected delegates over here. This is Aishwarya Nayak, pursuing B Farm second year KANAK MANJARI Institute of Pharmaceutical Science, Rourkela, Odisha. In view of the theme of the conference, I would like to present my topic, application of gene delivery in the and its gain in pharmaceutical research. Hope my work would be liked by everyone. So first of all, what is pharmaceutical research? It develops technique about the preparation and presentation of medicines. And in the next few slides, we would look into the new application of gene delivery, which have been coming upon in pharmaceutics. First curious question which would come upon is what is gene delivery? So the basic mechanism is the introduction of a foreign gene into a host cell, which would give us our required genetic requirement and also help in preventing various diseases, which the process can be seen in the following representation. Next, gene delivery system can be divided into two basic categories, viral transduction and non-viral transfection system. Transduction is delivering gene with the viral vectors, whereas transfection is delivering gene with chemical or non-chemical methods. So, in order to get an efficient vector system and to achieve high rate of cell transfection, we need to give upon the following limitation which I have pointed out in my presentation. A good gene delivery system should be effectively compact, transverse the plasma membrane easily, and deliver DNA to its target nucleus. Hence, more attention has been paid to develop non-viral vectors as an alternative for gene delivery. So what are nanoparticles? Non-viral gene delivery systems are plasmid DNA that are condensed into nanoparticles by a cationic polymer-based process, which we would see in the next slide. In this representation, I have shown the process of nanoparticle gene delivery, in which a cationic polymer nanoparticle is neutralized by a negatively charged DNA, and the end product give us the exact genome which can be easily encoded into the host cell. Next big question is, why nanoparticles? Nanoparticles can be considered as a good carrier for various therapeutic applications due to the following three reasons. They are easy to prepare, their size limitation is not there, and the cost of production is also very low. Other application of gene therapy in pharmaceutics are genetic markers for allele association, which is also helpful for knowing the effectiveness of the drug. Second is genetic probe testing. In view of complexity of information retrieval, computer-assisted searching by pharmacists is more compatible work. Next, we would see the development status of gene therapy. And first, in the first chart, we can see how cancer is very effectively treated by gene therapy. In second chart, we can see the global market for nanoparticles in biotech and pharmaceuticals have got an total compound annual growth rate of 22% in just 5 years. And the forecasted growth rate for the plasmid DNA manufacturing market report have got a CAGR of 14.52% from 2020 to 2027. This is the forecasted growth rate which we are seeing. So after letting to know the advantages and application of this high scope of this topic, can we point out the reasons to why gene therapy is still in a developing stage? So this answer is very valid. Gene therapy is still in a very developing stage and have many disadvantages which are being solved and coming up. So we hope that in near future, this would be the most emphasized topic to be studied in clinical studies. Thank you so much.
Thank you, Aishwarya, for your presentation. So any question to Aishwarya from participant? So I have the question. Gene therapy is started in India now. India started gene therapy.
So gene therapy has been starting, but it has not been approved by FDA. It has some disadvantages coming up, like it's because it's polymer based technology, right? So there are some disadvantages coming up which are being worked, but still it's like in cancer therapy, we are using gene therapy in cancer treatment.
Anywhere in world you have got any knowledge? Anywhere in world gene therapy treatment was started?
Sir, so I have read about it, but I cannot recall the name of the right now.
Okay. So thank you for your presentation. So with this, I handed over this session to my colleagues, Dr. Manoj Kumar Mahapatra, Associate Professor of KMIPS, to Mr. Mahapatra.
Oh, thank you, sir. Thank you, Professor Dr. M.K. Dalai, sir. So a very good evening to all of you, all the participants. I welcome you to this oral and poster presentation session of this international conference. So I would like to call Javanpreet Kaur, then Said Badri Mamul Mursalin, Dr. Minaketan Sahu, Mrs. Priyarasmi Chaudhary, and Ms. Anushka Das. So these candidates should be ready for their respective poster or oral presentation. So there is a time limit for 3 minutes to every participant. So first I would like to call Javanpreet Kaur. Please share your poster. I think she is not there. So next candidate, Mr. Said Badri Mamul Mursalin. Are you present? Yes, Said Mursalin is there. Mr. Said?
Yes, sir.
Yeah.
Yes, sir.
You can present your slides.
1 second, sir.
You can present your slides. Okay.
Yes, sir.
Yeah. Share your slides.
Is it visible, sir?
Yeah. Yeah, it's visible. Start your presentation.
Sir, myself, Sayyid Badra Imamul Mursalin, and I'm from Centurion University, Balangir, and I'm gonna present presentation on nanoparticle, nanotechnology, and nanomedicine. This is let's say nanoparticle. When nanomedicines are promising candidates, nanomedicines are promising candidates for targeting drug deliveries, especially at the cellular and molecular level. And this is a research article which is based on to provide a brief idea about medicines, nanomedicine. And then come to what is nanotechnology. Nanotechnology, according to the National Nanotechnology Initiative, nanotechnology is the understanding and control of matter at dimensions of roughly 1 nanometer to 100 nanometer, where this unique phenomenon enables novel applications, facilitating fabrication of devices on the nanoscale. And come to cellular.
Mr. Said, your voice is not coming.
Now,
Yeah, please put the mic near to your mouth.
hello.
Yeah, it's audible now. Continue.
Yeah. The nanomedicines, which is the medical application of nanotechnology, which shows immense potential to solve problems in medicine, especially in the cellular and molecular level. And the nanoparticles are the solid colloidal particles, which are ranging from 1 nanometer to 1 micrometer. And this consists of macromolecular materials in which the active principle is dissolved and entrapped. And come to the types of nanomedicines. Basically, it's based on the two features. One is structural feature and chemical. This is nano, and we can get polymeric quantum dots and up conversion nanoparticles. Then mechanism of interaction administration nanoparticles adsorption altering the
Your voice is cutting. Said, please adjust your mic. Your voice is cutting. Yeah.
Now,
Yeah, still voice is not coming clearly.
hello.
Yeah, now it's coming. Continue. Yeah.
In the next stage, phagocytosis of the nanoparticles by circulating macrophages leads to their
simultaneous elimination from the systemic circulation and accumulation mainly in the liver. And now come to surface modification. Surface modification is uncoated nanoparticles by and activate human any filter to with come to EPR effect. EPR effect is generally we can call the to per hyper the hyperpermeable vascularization. It is known as enhanced permeability and retention. Targeting and come to targeting method. There are two targeting methods. One is active and second is passive. And now come to preparation method of nanoparticles. There are 8 preparation methods. The solvent evaporation method, double emulsification method, emulsion diffusion method, net precipitation method, conservation method, salting out method, dialysis method, and supercritical fluid technology. And now come to nanoparticle characterization. They are particle size, polydispersity index, external morphology study, that is also we call TEM, and percentage drug loading, drug entrapment, and zeta potential. Now come to the advantages of this nanotechnology. Its intracellular delivery and targeting cell organelles, and improvement in intrinsic ADME profile of drugs, that is absorption, distribution, metabolism, and elimination, and enhancement of bioavailability, and also improve this is true.
Okay, Mr. Syed, have you finished?
Yes, sir.
Okay. So any question from the audience?
Mr. Syed, how can you measure the particle size? Syed.
So it may be from Coulter counter method, or maybe from electron microscope.
Okay. Then which instrument is used for this measuring polydispersivity index?
What, sir?
Which instrument you are using for this measurement of polydispersivity index, PDI?
Dr. Pollai, please switch on your audio. Video.
Audio is video. Okay. How can you measure which instrument is used for this polydispersivity index? Sahil?
I think he is unable to answer, so
okay.
Proceed another question.
Okay, thank you, Sai. So moving on to the next candidate, Dr. Minaketan Sahu. I think he's also absent. Mrs. Priyarashmi Chaudhary, are you there, madam?
Yes, sir. Yes, sir.
Okay, madam. Please share your poster and
Yes,
go on
sir.
your presentation.
Good evening to all. I'm Mrs. Priyarashmi Chaudhary. I am presenting about the poster presentation. 2 minutes of sharing.
Madam, please share your poster. Yeah, now audible, but please share your poster.
Sir, is it visible?
Okay, wait, it's
I'm
coming.
sharing, sir. Visible, sir?
No, not yet. It's presenting your screen only, not any slide or poster.
Is it, sir?
No. Keep the poster open, then go to the sharing.
Is it, sir?
No. I think share the entire screen.
Please open the file, then you share the screen with the file. Not that you are going to share your screen.
Ini present now. Kau bor kerja. Share. Is it visible, sir?
Wait. No, not. It is not coming. You are showing yourself only. Switch over to presentation.
If you are on laptop, then please present the entire screen.
Yes, sir.
So anyway, you call next participant if you are struggling.
Masih lagi?
Okay,
Dr.
madam,
Mahapatra, sir.
Yeah,
Dr.
I'm
Mahapatra,
yes.
please give some time to Priya, madam. Then kindly call next participant.
Okay.
No, not. You are not successful.
Yes.
Wait.
Coming. Okay, madam, you can go on. Now it's visible.
Coming?
Your poster is visible.
Make it to full screen.
Madam, please unmute yourself.
I think she take more time to present. So in meantime, you call next participant if possible.
Okay, so next participant is Anushka Das. Be prepared. Anushka Das, are you there?
Yes, sir. I'm there, sir.
Oh, yeah. You start your presentation.
Okay, you can start. Okay, madam, it's not coming. Your screen is only appearing. So please let the other candidate to finish.
Now let her stop sharing, then only she can enter.
Yeah, yeah. Madam, hey, madam, please stop sharing.
Yes, it is visible now. Manoj sir,
Yeah.
Sir, now I'm presenting, sir.
Okay, now you can.
Okay.
Anushka, you can
Sir-
wait. Okay.
Sam, now start, sir.
Yes, sir.
Okay, you can start.
Okay, sir. Thank you. Okay, sir. Thank you, sir. Sorry
Please switch on your video also, Priya madam.
Okay. Yes, ma'am. Is it, ma'am?
Yes, now visible.
Yes.
Please go on.
Okay, sir. Sir, my presenting is there.
Yes, poster is visible.
Yes, sir. Now, I'm presenting about a new paper, a new development in new liquid chromatography as UPLC. That is ultra performance liquid chromatography. It is a novel technique. It leads to shortening of analysis time and great savings in solvent consumptions. UPLC is one of the new developments in liquid chromatography. UPLC is based on Vendimeter statement, which describes the connection between linear velocity with plate height. UPLC contributes to improvement of these three areas, that is speed, resolution, and sensitivity. This is a new advanced category of the HPLC, which has the same basic principle and methodology with improved chromatography performance. UPLC achieved the above targets. Partners in 2004 launched and trademark ultra performance liquid chromatography. UPLC is a special version of HPLC, which has the advance of technological strides made in particular chemistry performance, that is system optimizations, detector design, data processing, and control. UPLC is based on the principle of these evolutions are governed by the Vendimeter equation, that is H equals to A plus B divided by V plus C into V, whether A, B, C and are constants and the V are the linear velocity, whether A are the independent of velocity and represents as ET mixing, and B terms is the represents as axial diffusion or the natural diffusion of the molecule, and the C is the kinetic resistance or equilibrium in the separation process, whether the kinetic resistance are there. In this time, like involved in the mixing form, the gas phase to the packaging, stationary phase and the leg back again. The greater of the flow of gas, the more of the molecule on the packaging tends to lag behind molecules in the mobile phase. Thus, the total terms is proportional to the V, that is velocity. Whether the UPLC system consists of some instruments are there, just as binary solvent, sample manager, including the volumeter, optional sample manager, form, detectors, other such instruments are there. In the difference between the HPLC and UPLC are, in EPLC, broader peak width provides resolutions. In UPLC have smaller peak provides better resolutions and more number of peaks getting more information. Whether there are less sample through the comparatively in HPLC, but in UPLC, higher sample throughout with more information. Such peaks are there in poster. In HPLC, sample injections volume is 20 microliters, whether in UPLC, sample injections volume is then 3 to 5 microliters only. Whether in HPLC, pumps operates at 10,000 PCM pressure, whether the UPLC pump operates at 2,000 to 6,000 pressure unit. As at last, describe about what is UPLC. UPLC increases productivity in both chemistry and instrumentations by providing more information per unit of work, as it gives increased resolutions, speed, and sensitivity for liquid chromatography. The main advantages is a reduction of analysis time, which also means reduced solvent consumptions. UPLC can offer significant improvements in speed, sensitivity, and resolution compared with conventional HPLC. Thank you.
Okay, thank you, ma'am. Any questions? So if no questions, then
Madam, your presentation is your presentation is your UPLC. What is its application?
Sir, again, please.
What is its application?
Its application, sir. Its application is now we will, as natural products, we will use as a ginger root,
UPLC.
which is the extraction of ginger root. UPLC and HPLC, as both are available same, because this methodology are the same principle and methodology instrumentation, but in some specific time will be there. That is the help in our UPLC will be now in days.
One thing from my side, one question.
Yes, sir.
How by increasing the velocity, can the separation be good?
Sir?
Because if you increase the velocity, particularly in this type of instrument, if you will increase the velocity, how the precision in separation will be good or ideal?
Sir, no ideas. Actually, I'm done with review.
Okay.
Okay, sir.
okay, thank you, ma'am. So moving on to the next candidate, Ms. Anushka Das.
Yes, sir.
So, madam, please stop sharing.
Yes, sir.
Okay, Anushka, you can present your poster.
Yes, sir. Yes, sir. So is it visible?
No. First open your poster, then share.
So now?
No. First open. First you minimize this. Yeah. Now it's coming.
Yes, sir. Good evening to all the delegates present over here. I am privileged to participate in today's conference amidst this pandemic era. So let me first introduce myself. Myself Anushka Das, and I am pursuing B.Pharm second year at KANAK MANJARI Institute of Pharmaceutical Science, Rourkela. As the lockdowns are being observed all over the globe, the national level pharmacy professions are performing as the frontline barriers. Pharmacists are globally providing services and also reducing the patient's burden on healthcare facilities. So coming to my topic, different approaches in novel drug delivery system, let me begin. The drug delivery system is a formulation that enables the introduction of therapeutic substance in the and it also improves the efficacy and safety by controlling the rate, time, and also place of release of drugs in the body. There are many ways to deliver the drugs, such as peroral, topical, transdermal, and inhalation. So coming to the advantages and disadvantages, it has many advantages, such as the dose is low and also it reduces gastrointestinal side effects, and it also has reduced dosing frequency, and also it has uniform drug effect. Now some disadvantages are dose dumping. That is, the dose dumping is a phenomenon where large amount of drug is controlled release formation and it is released, and toxic quantity of drug is introduced into the systematic circulation, and it also has stability problem. Now, coming to the routes of drug administration, it is basically divided into three main routes. That is topical, that includes skin and conjunctival, and enteral includes oral route, sublingual, buccal, rectal, inhalation, and also nasal routes. Now, coming to parenteral route, it includes subcutaneous, intramuscular, intravenous, and intradermal injection. As discussed earlier, the routes of administration, this is the market segment and the pie chart that I have represented, and where we can see that the oral control release occupies the major area, that is of 60%, and then comes the inhalation with 27%, and then the implants with 10%, and then transdermal part with 8%, and rest all others with 2%. Now, coming to the examples of NDDS, oral controlled drug release formulations, it occurs through the diffusion, dissolution, and osmotic pressure functions. Now, coming to the nasal drug delivery system, Navotix is a part that helps to reduce the migraine problem. Now, coming to the pulmonary drug delivery system, it has certain examples or machines such as nebulizers or meter dose inhalers and dry powder inhalers. Now, coming to transdermal drug delivery system, it has total 19 system, among which I know only estradiol, litodine, and pandamel, etc. And now coming to the mechanism, basically the mechanism is controlled with dissolution, diffusion, and the combination of both, and also osmotic pressure. So dissolution measures the extent and rate of solution formation from a dosage form, either it may be tablet, capsule, or ointment. It is basically for the improvement of bioavailability and therapeutic effectiveness, and diffusion is the movement of substance from a region of higher concentration to a region of lower concentration. And osmotic pressure helps to apply, osmotic pressure is the pressure that is applied to the solution to stop the fluid movement when a semi-permeable membrane separates a solute from pure water. Now, coming to the carriers, all these are the main carriers in drug delivery system, because they help in the improvement of solubility, stability, immunological breakdown, and also effect of enzymes. Now, coming to the conclusion, I would like to conclude by, based on pharmaceutical applications and available products, we can say that the above drug delivery system have definitely established their position in modern drug delivery system. Thank you. I would like to thank KMIPS for organizing this international conference on new horizons in pharmaceutical research. I am honored to present myself. Thank you so much.
Okay, Anushka, can you give an example of pulmonary drug delivery system?
Sir, pardon, sir?
Can you give an example of pulmonary drug delivery system?
So examples that I don't know, but I know the device that is used, like nebulizers and the metered inhalers that is used to.
Okay, in which disease it is being used?
So like if the patient is suffering from breathing problem, or if the lungs are being congested by deposition of water.
Okay. Any other questions from the audience? Okay then, thank you, Anushka.
Thank you, sir.
So, two candidates, Dr. Minakatan Sahu and Javanpreet Kaur, are not present. So I would like to request Professor Dr. Manoj Kumar Dharai sir to handle the next session. Please, sir.
So thank you, Mahapatra sir. Thank you for your control this session. Now I will name the next participant of this session. The name of the participant, our colleague Blessy TM. So now you present your presentation.
Thank you, sir. to be one of the most dreaded diseases. Although there are many types of cancer, they all start because of uncontrolled growth of abnormal cells. Normally, our cells grow, divide, and die. Because the cancer cells continue to grow and divide, they outlive the normal cells. Cancer is not one disease, it's a group of more than 100 different and distinct diseases. For this reason, the cancer treatment can include surgery, radiation therapy, chemotherapy, hormone therapy, and biological therapy. The doctors may use one method or a combination of methods. No one treatment can cure them all. So treating cancer is complicated due to various reasons like different physiology of people and the rate their bodies will metabolize drugs, the blood supply to the tumor that affects the drug getting into the tumor, and the physiology and the fact that this tissue can continue to change. Apropos to this, scientists are working on a new technology called nanotechnology that is considered with building things, generally materials and devices, on the scale of atoms and molecules. Nanotechnology, in combination with drugs or diagnostic molecules, improve the ability to target specific cells or tissues. These materials are produced on a nanoscale and are safe to introduce into the body. The nanotechnology provides rapid and sensitive detection rate that allows us to detect molecules even in a small percentage of cells. Nanotechnology has the potential to
Hello? Hello? Let's say your voice is breaking.
so maybe okay.
Now audible.
Okay, so this nano application of the nanomaterials include use of nanoparticles for imaging or drug delivery and nanotechnology approaches such as nanoparticles-based theranostics. Nanotechnology is a promising treatment option for cancer, which include innovative and non-invasive approach to cancer treatment. Thus, nanomaterials have particular advantages for cancer treatment and are being effectively exploited for improved delivery of chemotherapeutic drugs, resulting in both enhanced anti-cancer activity and reduced systemic toxicity. By this, I would like to conclude this poster presentation. Thank you. Thank you, sir.
So thank you, Blessy, for your poster presentation. Any question from participant? So I have one question to Blessy. Question is that how you cancer cells nomenclatured? How the cancer cells are nomenclatured? How the cancer cell are naming? What is the preferred process?
Cancer
Have any
cells
idea?
name means cancer cells are not cancer cells are
How
not limited to a particular cell. They spread.
No. The cancer cells are always naming they as for their origin. So based on their origin,
sir.
the cancer cells are named. Okay. So thank
Okay,
you your
thank you, sir.
presentation.
Thank you.
Okay. So now I like to call the last participant of this session, Smruti Malaika Datta. Smruti Malaika, can you show
Yes,
your
sir.
presentation right
Yes,
now?
sir.
Sir, this is so visible. Hello?
Yes, yes, it is
This is
visible. Carry
visible
on.
or not?
Yes, visible. Carry on.
Good evening, good evening, all of you present here. Myself, Smruti Malika Datta from 8th semester B Pharm. My topic is e-conference is cancer research technique. The research into cancer to identify cause, development strategy for prevention, diagnosis, treatment, and cure. This application includes surgery, radiation therapy, chemotherapy, hormone therapy, immunotherapy, and combined treatment modalized such as chemoradiotherapy. The main aim of the cancer research is the raising awareness of the disease and influencing public policy. Cause and development research into the cause of cancer involves many different disciplines, including genetic, diet, hormone, environmental factor, that is chemical carcinogenesis. The signs and symptoms are change in bowel and bladder habits, unusual bleeding or discharge, indigestion or difficulty in swallowing, a sore that does not heal. Many prevention are used, as don't use tobacco, protect your skin from sun, eat healthy diet, get immunized. Detection and diagnosis mainly done for the cancer are CT scan, bone scan, MRI, PET scan, ultrasound, X-ray, and most of these biopsy are done for the detection and diagnosis of cancer. Treatment are mainly used chemotherapy, surgery, radiation therapy, bone marrow therapy, immunotherapy, hormone therapy, target drug therapy, clinical trials, and the goal of cancer treatment have different objectives such as cure, primary treatment, adjuvant treatment, preliminary treatment. Cancer research techniques are focused on analyzing technique and protocol specific for cancer research. Some techniques are protein tyrosine kinase and phosphatase expression, profiling in cancer research and human cancer. Materials are used in cancer research technique are total RNA isolated from cell line or tissue, reverse transcriptase or fast strand complementary DNA, PTK and PTP amplification by polymer chain reaction. The alternative techniques are used for cancer treatment is acupuncture, hypnosis, massage, meditation, relaxation, and yoga. Patient and public involvement in research has been the focus or evaluating impact defined usually in term of participant. Practical contribute to enhancing research process. By contrast, there has been less emphasis on the perspectives and experience of those involved in PPI. Thank you, sir.
So thank you, Smriti, for your presentation. So
Thank you.
any question from participant? No. So I like to put a question that one point you say target drug treatment. What is it means?
Sir,
target drug treatment, you one point say in your slide. Target drug treatment. What is its mean?
Sir, it means particular one organ you can emphasize for treatment of cancer.
Okay. So with this, we conclude our session presentation. So before concluding, I like to thank all the presenting participation of this session. So they show their presentation with their views of the particular view. I hope they can explore more in their in their field. So I have observed many presentations. There is no reference in their presentation. So in future, they should their particular reference, because every information there is some reference. So you should go through the showing their reference always in their presentation. So with this, I like to thank all the participation of this session. So now I hand over the session to our Subhasri Madam to conclude any.
Thank you very much, Manoj sir. Now I would like to thank our both session handling persons and organizing director, Professor Dr. Manoj Kumar Dalai and Dr. Manoj Kumar Mahapatra for handling the session in a very nice way. Now I would like to thank personally to our chairperson, Principal Sir, Professor Dr. Vishwaranjan Behera Sir, for cooperating and encouraging in all aspects for conducting this international conference session. Now I would like to thank for the session to Professor Dr. Kunal Pal Sir, NIT Rourkela, as the first keynote speaker, and second keynote speaker, Professor Dr. Slawomir Wilczynski, head of the Department of Basic Biomedical Sciences, Medical University, Silesia, Poland, for their nice presentation regarding counterfeit drugs, and Kunal sir regarding the lipid-based drug delivery system. And I like to thank all the participants of this session, those who have elaborated and interacted with these scientific sessions like Priyabrata Panda, Abhishek Pradhan, and Amirnath Chatterjee, Aishwarya Naik. Then Dr. Minakatan Sahu sir is not here because due to the ill health. Next, Mrs. Priyarasmi Chaudhary and Anushka Das, then Blessy TM and Smriti Malika Datta for their very nice presentation and interaction on new aspects on pharmaceutical research. By this, we are going to conclude and we are going to end our this second session, scientific session of 19th February 2021. And again, we will meet on 9 a.m. tomorrow for the scientific session. And tomorrow we have to, the tomorrow session is very beautiful and encouraging and inspiring, because our Dr. Namrata Pathak Madam, DST, head and director of Department of Science and Technology, New Delhi, is going to connect along with Namrata Madam and different speakers from throughout the globe, whatever we have, to whom we have invited from India and also from outside country. Then all will deliver their talks and they will present, and they will we will get the opportunity to interact with them along with the participants, those who are going to present tomorrow. Thank you, sir.
Thank you, madam. Thank you. Hope for the best for tomorrow's session. And those participants and all staff members, please join early tomorrow.
Yes, sir.
So that's all, madam. Anything else?
No, sir. Now I am going to stop the recording.
Okay.